Notation. A great pleasure to introduce Graham Heimberg, who is a principal scientist at Genentech. Before joining Genentech, Graham was a postdoctoral fellow in Aviv Regev's lab, working on developing methods for single-cell analysis. And today, Graham will be talking about SC Immolarity, if I pronounce this correctly, a metric learning framework for cell type annotation based on a large curated compendium of published single-cell RNA-seq studies. With that, over to you, Graham. Very much looking forward to your talk. And if there are questions, I think Graham said we can ask questions during the talk, and folks can also post in the chat online.
Awesome, thank you. Hi, everyone. It's good to be back in Boston. So I'm going to talk a little bit about kind of a project that I started when I was here, but I think has really taken off at Genentech. And it covers a lot of ground. It touches on all the points Ludwig just mentioned. And so I'll get started with, I think, probably a really obvious statement, preaching to the choir here, that single-cell RNA-seq and single-cell transcriptomics is a really informative measurement. I picked just a few examples from past and present colleagues who have used single-cell RNA-seq to look at where genetics implicated genes are expressed in which cell types in macular degeneration in the eye, characterizing cancer development, and even finding new cell types that are disease relevant. And so you can do all these amazing things with this really high resolution measurement and many more. And also, I think another aspect about it that I really love is the scale of growth of the data. So as you'll see later, I've been collecting a lot of data sets, but just one kind of vignette from that is you can see when these studies were published. And if you look at that, the number of cells by the date, you can see actually that the growth rate of this data type is doubling every 6 months. So here I'm showing the number of cells. This trend holds roughly also 6 months for number of studies. So we have both a really high resolution informative measurement plus a really rapidly growing pool of data to work with. And so I think about how to put all this data together and to use it kind of in one cohesive way. I think there's a lot of reasons why I'd want to do that. With this kind of global view, we can try to find new cell state disease associations. We can look for when cell states are present in unexpected contexts, and we could even maybe relate in vitro to in vitro samples or cells, and much more. But this is just what I'm going to go through today. So I think this is a really important perspective that we can bring to our questions. And I think we probably all wanted to do this, but there's a few technical details. They don't feel like details in practice, but that keep us from doing this. These are all coming from different labs. We're all well aware with batch effects, and things like different labs are sequencing at different depths. They are aligning to different transcriptome versions. They report out their data in different data formats. So there's all kinds of reasons why it's not so easy to just put this together and look at it. But I think there is enough value in doing that, and so I'm interested in making new tools to address exactly this problem. And so that's where similarity comes in. I'm trying to use machine learning to learn a representation where I can relate all these cells from this exponentially expanding pool of data sets and use them together and cohesively. And so similarity is built on this concept called metric learning. And so metric learning is actually used I don't know if it's currently used. It used to be used by Google for facial recognition. And so how these models are trained, you feed it 3 data points at a time. The model takes in these 3, and you give it an anchor, a positive, and a negative. And the idea is the anchor and the negative, the anchor and the positive are examples of the same or similar data points, and the negative is something dissimilar. And so the model is learning features and an embedding that in your reduced dimensional space, the faces of the same person are brought closer together and faces of different people are pushed further apart. For this audience, this is one of my coworkers. So you guys may not know him, but internal to Genentech, the slide kills. One thing that is unusual and unique about metric learning, which I think is really interesting in this case, is that the models are trained so that the relationships between data points, the model is optimized about on relationships between data points. So unlike a classifier or an autoencoder where you take a data point and you try to minimize a reconstruction loss or get the class right, this model is taking in multiple points at a time and saying that how they should be related. And that shows up pretty clearly in the loss function. Just really simply here, we have the loss function about the anchor, positive and negative cell. And so we have the embedding of the anchor and the positive, and we have the distance between that, and we have the distance between the anchor and the negative. And basically we're wanting to relate them in a specific way. So for example, if the positive and the anchor are far apart, this quantity is very large, and the anchor and the negative are far apart, this quantity is small. So this is saying these dissimilar images, the similar images are far apart and the dissimilar images are close together. Then you get a large quantity here, and you take the max of a large quantity and 0, so you get that large quantity, and you add this to your loss function. So basically penalizing the neural network when this instance occurs. In the other example, when you have your anchor and positive close together, so these are the two Obama images, and Jason, my coworker, is out like an outlier, like he should be compared to the Obama images, then this quantity is small, this quantity is large, you get a negative number, you take the max of the negative number 0, and then you basically add nothing to your loss function. So basically, at that moment, you're telling the model everything's good, you don't need to do anything. So the idea that we had was, can we do the same thing but for cells? Basically, cells that we think should be similar should be close together, and cells that we think are dissimilar should be further apart. And I think probably a non-controversial statement is that cells of the same type should be closer to each other than cells of different types. So if you have, let's say, a T cell in the gut and a T cell in the lung, those are still more similar to each other than they are to macrophages. And so the idea here is that we're going to just take anchors and positives and negatives sampled based on this cell type identity and try to get the model to learn ways to put the labeled cell types closer together. And so you may have actually already used metric learning without even realizing. If you've ever used Google's reverse image search, you can take your image, upload it. It doesn't even have to be in Google, and they'll embed it, and they will look for these visually similar images. So we're actually going to do the same thing, or try to do the same thing, but for cells, where we can take a query cell, one of yours from your experiment, maybe the model's never seen it before, embed it in this common latent space with all the other experiments, and try to find the similar cells. Okay, so I'm going to go actually pretty quickly over the challenges, but feel free to stop me at any time. The three main challenges are, we want lots of data to do this. When you're working with author labels, that's going to provide actually quite a few issues. And then kind of a technical thing about the triplet loss alone doesn't preserve detailed information. So we're telling it to put all the T cells close together, but what about the activated state versus resting T cell and so on? And so, we want ways to preserve that level of detail. So, okay, anyway, first up, the data. One thing that people who know me might recognize, basically, I really like to scrape geo for data sets. I've done it a few times in my life now. And so, this is an amazingly rich resource. And so what I'm looking for is 10x genomics data. So that's coming from, that's a fairly standardized measurement, both in terms of sample prep and also ways that people are reporting data, like the file types people use to store that. I want it to be human data, and I want it to have the raw counts available. So I don't have to guess your normalization and reverse engineer or anything like that. So I want to be able to kind of control that. So that's a huge source of unlabeled data. And then for labeled data sets, we can look on CZI sub-hygiene. And also, of course, we all have kind of our favorite data sets floating around from different places that we can manually ingest. And so in total, this batch was 399 published studies. And you can see it really spans the whole human body. This kind of outer circle is different tissues. Then within that is different diseases. And then the innermost circles are individual studies. And so all in all, there's 399 studies, spanning this really wide range of different diseases, tissues, all kinds of conditions from the human body. And so, like I said, some of these have labels. And so you can see I'll be splitting these into the training and tests. And then largely from Geo is these unannotated studies. And so these are all going to be, these will come into the story later as kind of reference data set. When we want to do our cell search and we want to see which cells is this similar to, these won't factor into model training, but they will factor into potentially, into this reference set that we can search against. So that's kind of the data side. Another issue is different levels of heterogeneity, of granularity in author labels. So, for example, if you look at the cell ontology, which is what we use for our cell type labels, different people will be labeling cells at different levels of this cell ontology. By the way, the cell ontology does not look this pretty. So the problem is, as we create our triplets to train the model, we have to choose cells that are similar to each other, the anchor and positive, and we have to pick a cell that's dissimilar to each other. This leads to really ambiguous comparisons. Is a T cell and a Treg, are those similar or different? That's kind of an ambiguous question because technically the T cell could be a Treg, and the author hasn't labeled in that level of granularity. So what we do is we look for triplets, we sample these triplets, and we exclude any with this type of vertical ancestor-descendant or parent-child relationship. So for example, the T cell to CD4 positive T cell comparison would not be allowed, but the CD4 to CD8 comparison, that is easy for the model to say these are similar or dissimilar cells. And then lastly, this is maybe more detail than is needed at the moment. The triplet loss alone doesn't actually get us all of the way to where we want to go. So what we do is we add basically an autoencoder. So we have a mixed loss function of our triplet loss plus a mean squared error. And so we're going to train this model to do that kind of facial recognition trick, but also at the same time, try to preserve as much gene expression information at the same time. And so here what I'm showing is as we scan from just autoencoder to triplet loss and kind of blend these two loss functions in between, we can see different metrics that we evaluate the model on, and we can see kind of that it goes up and down. So the first thing what we're doing is we're looking at correlating our similarity metric from the space that we've learned to kind of a signature score. And so at this point, the signature score and similarity metric are matching best. And if we look down at the next row, here's a kind of a batch correction metric. And so I think kind of right here is a little bit of a sweet spot where we're scoring high on a lot of the metrics. So I can go more detail into this later, but just to give kind of a clue at what's under the hood is not just the triplet loss alone, I think is a kind of a key detail. Okay, so on to kind of the main story. If we have a similarity metric, if we can quantify how similar these different cells are using this metric learning framework, then we can do a lot of interesting things. We can visualize multiple data sets together without kind of an explicit batch correction step. We can classify cell types by just asking this unannotated cell, which annotated cells is it most similar to. And we can also do this kind of cell search of looking across all these studies to try to find the most similar cells. So first off, we can start to visualize these data sets together. Probably hard to see on the screen, but across all these studies, we can kind of slice it out by tissue, and we can show these embeddings, which have varying degrees of integration. But what's really cool, I mean, this is 63 blood studies and integrated, without any additional steps. And so just right off the bat, we actually get a pretty decent level of integration. Things like the brain look amazing, skin looks pretty good. So, across this, we're already starting to see these data sets coming together in this one common space. I think the visualization is probably the least interesting application. More interesting, in increasing order, more interesting is classifying cell types. And so this is actually a fairly straightforward problem once you have a measure of similarity. And so what we're going to do is we're going to take the pre-compute. So we have our labeled reference of almost 8 million cells that we trained the model on, and some that we no, we don't use the whole lot here, that we train the model on, and we can embed these all into the low-dimensional space by encoding them through the neural network. And so in the end, we have this catalog of cell types that we have their low-dimensional representation for, and we have the cell type label. Then we can come along with some unannotated query cell, and we can look, basically embed it and compare to our catalog and identify kind of which cells it's most similar to. And we can basically take that label from there. So on this end, we just use like a simple KNN classifier to say, oh, out of the 50 cells that it's most similar to, 49 of them are macrophages. Okay, this is a pretty clear instance that this cell is a macrophage. And the cool thing is this whole workflow takes 0.02 seconds per cell. And so you can start to classify large data sets, million cell data sets, scalably with this method. You don't need to input your reference data sets. We already have this huge reference available for you. And I think another thing to note is that we're annotating on a per cell basis. So I think a lot of papers generally annotate cell type at the cluster level. Then you have to pick number of clusters or cluster resolution. And anyway, so you are kind of dependent on that. I personally think it makes a lot more sense to be classifying single cells at the single cell level. And you don't need to choose the cluster resolution or any actually input. You can have a data set of one cell, we can classify it, and you can look at one cell. So I think that's really a nice feature of doing this. Yeah. Yeah, Hattie was first.
Hey, Graham. How would this handle cells that are not in your annotated reference, like out of distribution cells? Because even though they're amazing large data sets, for certain organs that are not deeply cataloged enough, there might be still rare cell types that may not be in your reference. So would it tell you that it's out of distribution, or is there a sense of the error or the uncertainty for the cell type classification if you were to, give it new data sets?
Yeah, so there is a way to measure that. So this is all based on a similarity metric. So basically, when we embed the cells, the distance between them is proportional, or inversely proportional to how similar they are. So if you say, okay, I have an unannotated cell and it's, I map it into this space and I compare it to the catalog, we quantify the distance that it is, or the similarity that it has to the catalog. So that is one way to check kind of, have I seen this cell before? It doesn't work as well as that in practice. I mean, yes, that does work, but the cells that we're missing are probably highly related to the cells that we already have. So let's say, I don't know, we were talking about B cells this morning. So let's say there's like a pro B cell or something. That might be pretty similar to a B cell. And if we don't have pro B cells in this catalog, it'll look fairly similar to a B cell. It'll be classified as a B cell. And so in practice, it works-ish. In theory, I think there's more to do here on getting really nice confidence metrics about each classification. Yeah.
Oh, yeah. Thanks. Hi, Graham. I had a very similar question. So thanks, Hattie. But just generally on this like for more background on this, so was the ontology, it seems like this is very sensitive to the ontology. So was there a process, or could you just tell us about the process of building out the ontology?
So the ontology is like a community source project. So a lot of people kind of define what the links in the ontology are. The only way that I'm using the ontology is just to get this ancestor-descendant relationship. So let's say the tree distance between CD4 and CD8 T cells is not an input into the model, just the fact that they are not kind of ancestor-descendant. So I actually think that we rely pretty loosely on the ontology just to get that comparison, just to make sure that it makes sense to say, are these cells similar or different? And that's it. We don't use a measure of how different they are in any step. Does that make sense? Any other questions? Okay, let me find. Okay, so cell type classification can work quite well. So let's see it kind of in action. So we can take, here's a kidney data set that is author annotated, and we're showing the embedding in our similarity space. And I'm showing the author annotations on this. So now we can come along and we can try to annotate this data set, and we can subset our catalog to just match the cell types that they are using. So we can do kind of a direct one-to-one comparison. And of course, this entire study is held out from our training set. So this is a brand new data set. This would be as if you collected a data set tomorrow and wanted to classify this data set. The model has never seen this before. And so we can classify and see how well it matches. And in a large degree, this matches quite well. And we can quantify this in a confusion matrix, and we see our nice diagonal here. And we can also zoom in to places where it doesn't match so well. So for example, these CD4, CD8 T cells are being, a little bit of mismatch between my predictions and the author labels. So we can kind of take a closer look at these cells. So what I'm showing here is just subsetted down to these cells that are like where the confusion matrix is. And we can see the author label cells and the similarity prediction. And basically in the similarity prediction, this top cluster is going to be classified as CD8s and the bottom is CD4. And so we can take CD8 and CD4 gene expression, and we can see largely on in the top cluster for CD8 and largely on in the bottom cluster for CD4. So I think in a lot of cases, we're seeing these examples of where we can potentially classify even more accurately than the authors. Probably in this case, this was at their cluster boundary. And, it's a hard distinction to make at the resolution that they were clustering in. In other cases, actually, I don't show this here, but I said here that we restrict to the labels that the authors use. We can take away this restriction and use the full catalog, and we can often get more granular resolutions than they do. So we find in this data set, we find Tregs, we find different subsets of fibroblasts and all kinds of stuff. The authors are correct, but in different cases, there's few pockets of cells that have an even more appropriate fine-grained annotation. So I showed you this for one data set, this young data set here, but like I said, I held out 14 data sets altogether. And so we can kind of look at the performance across each of these subpopulations per data set. So each dot is one cell population. So let's say author annotated CD8 positive T cells. And basically, I'm asking how well do I match the author annotations when subsetted to their set of cells. And some cases we do better than others, but you can see largely the density around this is above 80% usually. And there are some data sets that we do pretty poorly on. And so in this case, this is an interesting example. This is a data set of CD4 T cells broken out into naive, memory, and these really granular subsets that clearly we have a hard time doing. I would actually maybe argue the authors also have a hard time distinguishing these. But in general, these are hard distinctions to make. And so maybe they're doing better than we are, but no one's really doing great here. Yeah.
What are the dots?
Each dot is one cell population. So that would be one author label. And I'm quantifying how well we're classifying that cell type class. So these classes are very tough to distinguish, and we don't do such a great job on them.
So comment refers to the questions earlier as well. One of the things that you might be able to do to help out is to give sort of a distribution of within cell type distances for the author or for your embeddings, so that you sort of, the 50 closest matches were all CD4 positive T cells, but they weren't really as close to that group as that group is to each other, right? You're sort of, part of what gets used here a lot of times is totally ignores the fact that you actually have within sort of cell type distances that would help inform you about the new ones.
Yeah, absolutely. And I think, this is not exactly a question, but in the extreme example, if somebody puts in a cell, let's say a random profile, the model's going to try to stick it somewhere. And inevitably, it's called a hepatocyte. So sometimes I'll get questions from people like, why does my brain data set have so many hepatocytes? And so, we need to put in a few more kind of confidence and quality kind of control metrics on this to show like, okay, the model's just sticking it somewhere. It's closest to hepatocytes, but it's in reality, it's nowhere near the hepatocyte region. And so, that's an error. So we definitely need to be adding in more of these, but it's pretty new. It's a new thing. So it's hard to get that right. And we're still exploring that, but definitely a work in progress. Okay, so then lastly, I'm just taking a tiny fraction of the 23 million cells here, but we can sample from all of these data sets and start to put them together into one picture, and we can classify all of these cell types. I do it more granularly than this, but you can't show 120 colors on a UMAP. But the idea here is this starts to create a really nice starting point for a lot of downstream analyses. That we have all of these different tissues, all of these different things in the same space, and we can classify by cell type. Means that we can now slice by tissue, we can slice by disease, we can slice by cell type, or any kind of intersection thereof. And just one kind of last look at cell type classification, we can see, using integrated gradients, basically a way to look within the neural network to see which features, in this case, which genes are most important in driving the embeddings and therefore classifications, we can see which genes kind of are most important for each cell type label. And so what I'm doing here is I'm taking the single cell GTEx differential expression. So basically they're taking their each of these carefully annotated cell types and doing differential expression to find which genes are kind of most unique for these different cell types. And you can see that in my kind of gene list, many of them are at the very front of my list, and it flattens off, meaning that the later genes aren't that important in either of our lists. I would say we do pretty well in everything except the neuroendocrine cells. I don't really know what's going on there. But this is encouraging to see that, you know, the classifications make sense, we get a good degree of accuracy, and the model is making decisions based off of genes that we know and love. And so the last, and I think the most interesting application here, is to search for similar cells across studies. And just like the cell type classification, we're going to use kind of a similar framework where we have, in this case, our full almost 23 million cell reference database. We can pre-compute, we can pre-embed everything. And then we can take a query cell and embed that in the same space, and we can try to find which cells are the most similar to that. So gonna kind of apply this to a vignette that I work on at Genentech and work with Jason, the guy who was not looking like Obama. What they were working on before I even got there was looking at IPF and interstitial lung disease. So in this case, IPF and systemic scleroderma. And they did a bunch of single cell RNA-seq to profile these lungs. And in their clustering, they found this one cell population, or two, but we'll focus on this one, that is really expanded in disease. And they see it essentially correlating with disease severity. So these FNs, or fibrosis-associated macrophages, you know, are all of a sudden of interest. What are these cells doing? Are they protecting the lung against further disease? Are they driving fibrosis? So what we can do is we can apply this framework to these cells. So we can take these FM3s, basically reduce it down to the typical FM3 profile, or like the centroid of that cluster. And we're going to treat that as our one cell in this case, and we're going to go to try to find similar cells to that. So what we can do is we can quantify how similar each macrophage or monocyte in our entire collection is to our query fibrotic macrophage. So that's what I'm showing here. Each column is a different tissue sample from our collection. So we have 3,500 different tissue samples that we're looking at that all contain monocytes and macrophages. And then on the y-axis, I'm showing how similar these cells are. So how close together in the embedded space these cells are. And you can see on the left, these samples have very little similarity to the fibrotic macrophages that we started with. And on the right, you can see some of these samples have, you know, more right-shifted distribution. And so we see some of them definitely having these fibrotic macrophage-like cells. The cool thing here is with all of these cells, with everything pre-computed and with this model and with this representation, this comparison takes 2 seconds. So we can be doing these routinely for all different cell types. You say, oh, I actually want to include a few more cells in my fibrotic macrophage, you know, prototypical profile. Like, no worries, we can redo that in 2 seconds.
2 seconds on what kind of machine? Your laptop or?
No, it takes a bit more RAM than that, but I mean, it's nothing too fancy. So probably about 64 gigs of RAM. If you have access to 64 gigs of RAM, then you can do this in 2 seconds.
MacBook Pro.
And actually, if 2 seconds is too slow, so what I'm showing here is an exhaustive comparison, taking this prototype fibrotic macrophage and computing the distance to every single monocyte or macrophage in this collection. But if you just want to do an approximate search, so like, you know, take 1,000 nearest neighbors, that takes a 20th of a second. So if you have your choice. You can do the slow version or the fast version. But so we can start to ask questions that start to shed light on what these cells are doing and how we can study them. And I think really simply is, you know, where else are these fibrotic associated macrophages? And I think to me, it goes kind of in 3 levels. First, are they in other IPF or interstitial lung disease data sets? Because if they're just in ours, maybe it's some technical artifacts, I don't know, probably not worth developing a drug for. And then another question is, we still don't know exactly what, we have hypotheses, but we don't know exactly what role these play in fibrosis. So are there instances of fibrotic diseases without these cells? Or do they appear every time there's fibrosis? And then lastly, the way to study these right now is we wait until we get human lung samples from ILD patients from a lung transplant. And that's, you know, comes and goes. So, you know, it'd be really nice if we can find a nice model system to study these in the lab to learn more about functional kind of role in disease. And so to do that, we'd like to see, are there any in vitro cells that actually look similar to these? So we can take this previous picture and we're going to just do some very simple stuff. We're going to count where the most similar cells are. So what I'm showing here is just the 95th percentile. We could take all the cells above the 95th percentile line and just see which data sets do they come from. So back to kind of this study and tissue and disease view, we can see, you know, which conditions give rise, or which conditions these cells are present in. And so you can see, obviously, and they're quite common in the lung. Here's the interstitial lung disease. And what you can see is that quite a few of these are actually in ILD studies. So at about 9 out of our 11 studies, we see a solid number of these fibrosis-associated macrophages. So question number 1, I mean, these are definitely not just in our data set. These are in other people's data sets. To be honest, this is what we expected. Other people have talked about cells like these, but they use different markers, different nomenclature. It's a little unclear if we're all talking about the same thing or not. So this is kind of a nice starting point. And we can pull out one individual data set and we can see kind of where the most similar cells are. And you know, we can separately score them with our favorite genes, and we can see that these are talking about the same thing. We can also look and find them in places we didn't expect. So pancreatic cancer, we have 2 pancreatic cancer studies in this collection, and both of them have fibrotic macrophage-like cells. Maybe not entirely surprising, given that this is a highly fibrotic cancer, but it's cool because we didn't start out looking there, and this result essentially comes, you know, in 2 seconds now. So that's kind of a nice thing to see and a nice thing to you know, explore further. Obviously, you can pull out these individual PDAC data sets and explore them kind of to your heart's content. This is just a starting point here. And then lastly, we can see where these are in, or if there are any similar cells in these in vitro studies. So what I'm showing here, everything that I showed previously was primary tissue samples. Now I can do the same thing, basically just subsetting the reference to in vitro samples. And we can see kind of, again, a range of similarity. And we can see that, you know, the far right, these top 2 actually come from one study, as well as these 2 down here. What's really interesting about this, at first, actually, I didn't really know what to do with this. We kind of ignored this for a while, but then realized the value of having kind of a potential in vitro model for these cells. I think the most interesting thing is that this is a study we would have never, ever looked at. The original paper where they did the single cell RNA-seq, they're trying to expand hematopoietic stem cells from PBMCs. They put PBMCs into a hydrogel with a bunch of cytokines and growth factors, and they do expand hematopoietic stem cells, and they assay that by single cell RNA-seq, and they publish their single cell RNA-seq data, and they share that to show that they've expanded hematopoietic stem cells. This is completely unrelated to interstitial lung disease. These are not from lung. They're not studying lung disease, anything like that. But what we see is they have this time course in their system, which is awesome. And you can see their starting point has cells that do not look like our fibrosis-associated macrophages. And then at day 5, they look significantly more. So kind of we're looking in this region down here. There's nothing really here on day 0. And then there's a lot here on day 5. So we've gone and replicated this ourselves in a mostly same experiment. We took it out to 8 days. I actually don't know why. But you can see we actually can also replicate these findings and get cells that are similar to the query fibrotic macrophage. So that's pretty cool. And we can take it down kind of to the last step of looking at our favorite genes. That before I started on this is how we used to define the fibrotic macrophages. And you can see day 0 has very little to no expression of these genes. Day 5 has quite a bit more. And we see that kind of replicated in our validation on this day 8 time point. So we've shown now that you see them across ILD studies and other fibrotic contexts, and actually we have a starting point for an in vitro model for these cells. So that's pretty much it for the results. But I will say, I think using all of these data sets together has really changed how I look at single cell RNA-seq data. I think it's really exciting to take hundreds of public studies and squeeze all of that information out and apply that to my domain, whether that be a rare disease that you don't have much information for, or ILD that we actually have a ton of data sets for, actually. It's still really cool to be able to connect these data sets and pull information out of them. And so, I mean, that leads to the cell type classification for millions of cells. We can do these queries, and we can do these really large visualizations, across these data sets. Another thing I mentioned earlier, I mean, slicing by cell type or cell state, now it becomes trivial to ask, which cells in the entire body is my gene expressed in. And so, that becomes, I think, a very valuable question, especially when you're developing drugs and you want to find and validate a target. You want to make sure that it's where you think it is and not where you don't know. So I think those types of things will become increasingly common, increasingly routine at a pan-body scale. I think that my analyses have become much more fluid. So the embedding works on the per cell basis. And so if you want to subset that, you can subset that. You do not need to recompute anything. If you want to add in a new data set that you just collected today, you don't need to recompute this embedding. This model is trained to be general. So in theory, I hope that everyone in this room wants to go use it, but no one actually has to retrain the model for their specific case. I think there maybe are a few exceptions, but largely, for a COVID study, there's plenty of COVID data in here. Probably don't need to do anything additional on this. So I think that's actually a really interesting thing that you can kind of combine and change the scope of your study on the fly. And then lastly, I think one of the main things that I'm looking for now, people ask about, oh, what's your threshold for similarity of cells or stuff like that. And I'm moving more away from, oh, I'm looking for a similarity greater than 5, and trying to now see, okay, do you actually see it across 2 of 3 cancer studies, or 7 of 11 ILD studies. And so I think it's really cool to move to replicating cross studies because, and that's, I think, the end goal when you're trying to get confidence in your results anyway, that you should see this signal. When you see the signal, you should also see it in another data set that is, the same context. So I think that's something that is really interesting, and I'm enjoying actually coming around to this because choosing a threshold is no fun. So lastly, I think trying to publish this any week now, and so it'd be awesome for people who want to play around with it. The code will be open. Pre-trained models will be released under non-commercial licenses, potentially open. And so that means that, we'll have demo notebooks that you can take, your data set of interest and bring it into the space and do your 2-second query or 0.05 second query, whichever you want. And you can try it on your own applications. And yeah, with that, we have a nice API and demos. So we already use this within Genentech, and we have workshops where we train people and they're using it the same afternoon on their own applications. So I'm looking forward to releasing that out into the world, and hopefully you guys find it interesting. And then of course, there's actually a lot of people that went into this project. And so all of this row have done a ton of work or advice or testing or software development on similarity itself. And then we have Daryl and Toby who were doing all the fibrotic macrophage work on the side and have been testing the models and play with it and tweak it. And then I didn't really get to talk about it much. I just kind of mentioned for the cell type markers, there's kind of a whole interpretability component to see which genes are driving how the neural network is embedding the cell, which genes are most important. And so that's from some machine learning colleagues. So yeah, with that, happy to take any questions.
Well, thank you so much for this super interesting talk, Graham. Yeah, I think if people have questions, I will go around with the microphone. Maybe I start off with 1-2 questions.
Sure.
So of course, given how transformative the method is for you, you have talked a little bit about availability. Just wondering, it's implemented in Python or? Yeah, so you planning on making this available, something like a Python package or something?
Yes.
Yeah.
So we actually already have a nice API that we're using, Python API that we use internally. So when we get the green light, we will share that. It's ready to go.
And the data compendium itself, this will, we learned from Hector, not come super soon, right?
So the data is a little bit of a different situation. It's all public data. So, I mean, there's a few ways in which we'll be releasing it, but we're not going to repost all of the original data. So we'll have a list of the data sets that we use. So either you can go find them. The other thing is release the pre-computed embeddings for all of the data. So you can do the queries without having to go download the data set. Then maybe you'll find, oh, my cells are, there's a lot of similar cells to my query cell in these 5 data sets. So you can go download those 5 data sets if you want to do kind of a deeper dive. So we'll have the list, the pre-computed embeddings, and the the pre-computed k-nearest neighbor index. So you can do the 0.05 second search.
And the second question that I had, so for the cell type annotation tasks, there are obviously already a bunch of methods out there, like single RSL tables, to just name a couple. Did you look at all into comparison there? How a similarity compares to those in particular in this withheld experiments that you do there?
So I didn't compare them. I didn't do a lot of benchmarking against these existing methods. I think this is a slightly different use case in that you don't need to go collect a reference data set that you're going to match to. We could benchmark kind of that same use case here. But yeah, I think
Or
My-
do you have like an intuition what works better here?
honestly, I would probably say, actually, so I didn't do this benchmark. Someone else looked at cell typist. And in this specific use case, in this specific application, they came out the same. My intuition is they're going to come out the same. I think, though, the reason I use this is I don't need to go pick a reference. It's super fast. I don't need to cluster the data. I don't need to do anything. So it's really about like out of the box usability that I think. And in the end, the best annotation, I mean, as we talked earlier, if you really want the highest quality annotation, I would start with this and then go to kind of an expert, to sit down and manually curate with them. So we released these annotations in Genentech, but we call them cell type hints, so that I'm not like liable for any issues. So I guess I would kind of suggest the same. You can use this to get cell type hints, and that's a really good starting point. And you can choose where to go from there. And I'd probably say the same for other people's methods too. I'd maybe call them cell type hints.
All right, other questions here. We also have questions online. Maybe we start some questions.
Beautiful talk, Graham. Oh, sorry. You can go ahead. Yeah, the online question can go ahead. Yeah.
Okay, can you hear me?
Yes.
Yeah, sorry for not coming back to the seminar room. I have a question. I wanted to make a call back to something that Rob said, who I assume is in the audience this morning. He talked about how he thinks that a lot of the future of drug discovery lies in phenotype discovery. And I was thinking about this fibrotic associated macrophages that you talk about, and I think it's a fascinating discovery. And something that even visually I thought about is that it kind of sets a kind of a spectrum of individuals based on something about their cells. So in a way, it made me think that this could lead to some interesting kind of like individual embeddings, kind of like, for example, within COVID patients, do you find like clusters based on like features like their percentage of macrophages that score above something or something like that? And I wonder if you've ever thought about that.
Definitely. I'm very interested in the individual level embeddings, but I think I haven't quite come up with a way to think like a framework to get that in a useful way where I'm going to actually learn something from that. So yeah, I keep thinking about that. If you have suggestions, let me know, please. Because I mean, there's tons of data and we now have a common space to consider all these cells in. And so I think that's a good starting point for a tissue sample level embedding.
Great talk, Graham. So I was curious if you've thought about this concept of meta cells that some people are thinking about and whether that can help identify, rather than this continuum of cell states that we observe in single cell data, which is probably an artifact of our measurements noise and et cetera, if there's any kind of like basis for distinct like sinks of cell states that a given cell type could occupy across different disease or different biological conditions. And somehow if there's a right way to group cells within each cell type using this meta cell idea, and perhaps have a data-driven way to say, oh, these type of T cells have these distinct like source, or sorry, like, I guess like these are the possible states that we could assign names to in a way. I don't know if that makes sense.
Yeah. I mean, I guess it's about kind of resolution. When we look at all of these cells, what resolution do we have? And the idea of meta cells is anything that we don't have the resolution to discriminate, we consolidate that into one. Have I thought about that here? Yes.
Or like if it's possible to relate to different types of functions that a cell can perform and then link that to this meta cell idea.
In my ideal world, I would go in a different direction to address this problem, which I understand, like I appreciate the issue. I would go probably in a different direction of trying to add in data augmentations when I'm training so that the model tries to, so basically I can tell the model which shape the noise is in to try to reduce that. So in my ideal world, I'd actually, Metacells actually uses like a sampling model. And if the cells are close enough together, basically you can't tell them apart by sampling noise, then it consolidates them. I think then maybe what if we just put sampling noise on the training of the same cell, we see the same cell augmented a few times, and that way the noise gets kind of baked into the model. I haven't thought of a way of consolidating beyond that. I mean, I wanted to do some kind of noise canceling, and we all want stuff to do noise canceling. But that's kind of my best thought at getting kind of noise canceling. I don't have any kind of grouping beyond that. But separately, I have thought about switching all of this to Metacells, because while the queries go super fast, the thing that takes a long time is actually loading in basically the library, which has all kind of these big files behind it. And so what I show now, I mean, this is the publication version. We have bigger references now that we use, and it's up to about 3 minutes to load the Python module, which is kind of annoying. Once you do that, then it's super fast. But if this is really doubling every 6 months, I'm not gonna let that load time double every 6 months. So in the future, I can't afford to keep every single cell, at least in this current framework.
Just a plea not to put a non-commercial license on it. You will exclude pretty much everybody that works here and probably everybody that works almost everywhere.
So this is an ongoing discussion at the moment, but you should expand on that so I can bring that back and say why we shouldn't do that.
Because it's not a well-defined term, right? And so what does it mean if I work on a project like one of the ones that we talked about this morning, which some of the data came from a pharma, right? So how's that not
interesting,
non-commercial? How would it be if I went and gave a short course where I mentioned this in the middle of it, but I charge for the short course, right? That's commercial.
Got it.
There's just no way that anybody who's functioning in the world that intersects between academia and industry
is going to tailor each.
You just can't touch these things because you can't afford to have them in the middle of your workflow.
Okay, I'll bring that back. I mean, personally, why not open? So
there's another question online from James Weber.
Yep.
Hello, Graham.
Hi, James.
Nice to see you. There's a lot of friends in the chat here. I was actually, this kind of touches on the previous question, which is, you mentioned building noise into the training. And so I was curious more about the training process, and it sounds like you have an autoencoder in there, as well as the metric learning. And how much do you think your model is based on the structure of the autoencoder, or how that works? And is it worth, I understand a really big benefit is you don't have to retrain this huge model, but like maybe you want to. Like, is there a benefit there, you think?
Okay, wait. I feel like there's a couple questions.
Sorry, let me make a shorter version of this question. Okay. Do you think that you could, knowing now that you've done this, build a better model and for the noise and for the single cell data?
It's not that something immediately obvious pops out to me on what a better model would be. However, I would say that one direction that I would look at is this is training only on the author annotated data, which is about a third of all the data that we have. So I think in version 2.0, let's say, if that's what your question is, what would I do next? Version 2.0, I think I would try to use more of the unannotated data, whether in pre-training or somehow change kind of the training, like the model type to use all of that unlabeled data as well. Does that answer the question?
Yeah, that makes a lot of sense. So we can talk later.
Great, so I think last question.
Hold on. Okay. I'm curious, you got really good inference times. So exactly how big of the weights are you working with, or the one that you're willing to publicly share, hopefully very soon?
I mean, this is actually, I mean, the rest of machine learning is really good at this. So they have these tools. I mean, these are approximate KNN libraries that exist off the shelf. And so you can Google that, and people will benchmark their KNN times. And so you can just pick today's best, which is probably better than the one I used. But people use it for all kind of domains of machine learning, information retrieval type of stuff. So the key here is actually getting the problem formulation into a KNN compatible question. Once you do that, then everything moves very quickly because you're just taking everyone else's tools. And the key here for that is making the distance in latent space mean something, by design. And then if the distance in latent space means something, then a nearest neighbor search is really informative. And that's where we have really great tools.
So a key is then just in the weight of vector space of it being embedded, or is it just?
Yeah. I mean, and there's no secrets there, but the idea is that the nearest neighbors in vector space are the answer to your question. And the nearest neighbors are there's very efficient ways in finding nearest neighbors.
So you're just using a regular KNN just to find metric distance.
Yeah, exactly. Yeah, nothing,
Okay.
No secret sauce.
Okay.
Well, then, thank you so much for answering all these questions.
Yeah, of
And
course.
really, thanks again, Graham, for this fantastic seminar.